My name is Jae Lee. I'm a professor here at the Miami Project to Cure Paralysis, and my lab investigates the wound healing process that occurs after spinal cord injury, including inflammation, cell proliferation, and tissue remodeling that occurs at the injury site. Of the research that's going on in my lab right now, what excites me the most is that over the past 12 years, we've been investigating the basic biology of how all the cells at the injury site interact with each other during the wound healing process. But now we have the computational and the bioinformatic tools to really help us try to understand how all these complex processes are occurring, so that we can then use that to develop drugs and other therapeutics to help people walk after spinal cord injury. Well, the research that's going on right now, I think how we can help people with spinal cord injury is that first, it's going to help us understand how the cells are interacting at the spinal cord injury site to help us understand all the complex interactions of these cells. There are over a dozen cell types that are interacting with each other after spinal cord injury. They include cells that are endogenous to the spinal cord, but also immune cells that infiltrate because now the blood vessels are ruptured and all these immune cells are coming in. And these cells interact with each other in complex ways that we're just beginning to understand. But now we've been investigating this process over the last decade or so. And the goal now is to help get computational tools and bioinformatic tools to help us to take this to the next step, because these interactions are way too complex for at least my mind to grasp. And so then how can we use computers, for example, machine learning, bioinformatics, artificial intelligence, to help us develop therapeutics? And what I think how this can help people with spinal cord injury is that these computational tools can translate kind of the language of the cells, for example, its genetic signature, right? That can be used kind of as a fingerprint of what these cells are and how they function, so that then the computational tools can help us to develop individualized therapeutics that target individual cell types, because one drug that might help one cell type might be detrimental to another, right? So how do we use computational tools to develop more precise therapeutics that can target not just the cell, but a biology of that cell that is detrimental to gaining function after spinal cord injury? That is, I think, how my research can help people with spinal cord injury. So what triggered my path to science was that when I was an undergraduate doing research, I used electron microscopy to look at chick cerebellum development. And images of looking at that, it just opened up my eyes to really a whole new world. And that is what actually still excites me to this day, is that when I look under a microscope and I see a tissue of the spinal cord injury site, I know that the answer is there somewhere, and we just have to find it. And it's like looking into a whole different universe. And I'm seeing something today that no one in the world has seen before. And that is what's most exciting to me and what has led me into science in the first place. My path to the Miami Project was a bit unexpected. I've been in the spinal cord injury field for a long time. I investigated spinal cord injury as part of my PhD thesis, did my postdoc in spinal cord injury. So I've been in the field, but what led me here was really the collaborative nature and the family-like environment of all the faculty here. When I interviewed here, there was a level of energy and enthusiasm, not just of the faculty members, but the trainees, the people on the first floor. They were all so excited about what's going on here, and the future about what's to come at the Miami Project is really what drew me here.